Welcome everybody to the last OEH seminar for this semester. And please join me in welcoming Bianca Malouf, who is a current master's thesis student in the School of Population and Public Health in the OEH theme. She holds a bachelor's degree from McGill University with a major in biochem and minor in environment. And Bianca is a recipient of the WorkSafe BC Research Training Award 2011. So today she will be talking about her thesis titled An Airway Oxidative Stress Due to Diesel Exhaust Inhalation in Humans, Analysis of Exhaled Breath Condensate. Thank you.
Hi everyone. Nice to finally be presenting at this meeting. It's an honor, so hopefully you guys learned something, and I as well from your comments. So we're going to go through these components of my thesis and hopefully at the end have some time for questions. Briefly, diesel fuel combustion releases 100 times more particles per distance traveled than gasoline fuel combustion. And this exhaust profile is mostly ultrafine particulate matter that has been linked to asthma exacerbation and pro-inflammatory responses. This is particularly problematic since the WHO released in 2005 a report that there'd be a 20% increase in diesel fuel consumption by 2020. And this, despite the very effective control technologies to control diesel exhaust, the replacement rate of these control technologies on the modern workplace and in the cities is quite slow. It will take several years, if not several decades, to decrease all emissions to zero. So controlled diesel exhaust studies have shown increases in airway resistance, increases in inflammation in healthy individuals, and bronchial inflammatory changes. Now these are both in healthy individuals and asthmatics. Asthmatics also have lower levels of antioxidants than do healthy individuals, and I'll explain the importance of this in the following slide. So what is diesel engine exhaust? Basically, as most of you know, diesel exhaust is a solid carbon core with elemental and organic carbon at the center, in which there are trace metals, and in which volatile organic compounds basically adsorb and desorb from the solid compound depending on the environment in which they're emitted. Also with diesel exhaust comes gases such as CO, CO2, NOx, and SO2. And what happens with these is because most of them are small particulate fraction, it goes deep down into the alveoli layer, crosses the cells, and into the systemic circulation. And diesel diesel particles act in two ways. Basically, they can interact directly as reactive oxygen species with the cellular components of the lungs, or they can induce genetic modification or signaling pathways that will cause the release of cytokines and other inflammatory markers by actually activating cell signaling pathways. So why is this problematic? Well, for asthmatics, they are doubly affected by diesel exhaust in that they have lower oxidant defense systems, so they have lower levels of, antioxidant enzymes, and they have lower levels of antioxidants in order to combat the oxidant effects of diesel particles. So how do you measure most of these health effects? Typically, controlled exposure studies have used bronchoscopies to measure changes in respiratory health. These methods are very invasive. Other methods, such as sputum inductions, which are semi-invasive, have been used. And in our study, we used exhaled breath condensate, which is a very non-invasive, easy to use, and reproducible method of obtaining a respiratory fluid in order to measure outcomes. So basically, as you can see, you can do it with children, you can do it with adults. Individuals just breathe into a propylene tube that has an aluminum cooling sleeve on top that was cooled at -20 degrees Celsius, and it condenses the breath that you exhale. Exhaled breath condensate. It's very straightforward. And what happens is the lining of your airways your areas are kind of covered with this small layer of fluid that becomes aerosolized into your into your breath when you exhale, and this is thought to reflect the underlying health of your lungs. So what we do is we analyze exhaled breath condensate for several different markers in order to assess, how are your lungs doing. So this has been used for COPD, asthma, cystic fibrosis, etc. So with the interest of our study, we looked through literature and identified several biomarkers of interest, two of which I will spend a lot more time focusing on in this presentation. So the Montucci group extensively researched exhaled breath condensate levels of 8-isoprostane, and what they were able to show is that exhaled breath condensate levels of 8-isoprostane significantly increase with increasing asthma severity. And so this marker is actually a good marker of airway oxidative stress or it's a good marker for diagnosing asthma, and it's a reliable marker of exhaled breath condensate levels of 8-isoprostane. Sorry, a little bit repetitive. I think one of the most important studies that we use to model our results, or to model our analyses on, was exhaled breath condensate pH. Was a study by McCreener in 2007 published in the New England Journal of Medicine that looked at basically ambient exposures of diesel exhaust. So what they did was they took 30 mild asthmatics, 30 moderate asthmatics, and asked them to walk for 2 hours either in a park or along a busy street. And they wanted to identify what airway changes happened between those two exposures. And what they saw was a significant decrease in exhaled breath condensate pH, so a significant acidification of the pH 3 and 6 hours after exposure. And this is really interesting because this use of exhaled breath condensate pH was one of the most significant findings at the time. So another way, another component of that we were talking about was antioxidants and levels of enzymes in the system. And what was found is that in vitro studies using N-acetylcysteine to as a supplementation against diesel exhaust exposure of cells showed that oxidative responses were suppressed by the use of N-acetylcysteine. In animals, a sister compound, N-acetylcysteinamide, reduced oxidative stress and inflammation caused by diesel exhaust. And then most importantly in humans, this was a novel finding at the time. Ramirez in 2002 showed that in children in Mexico that were exposed to air pollution, supplementation with antioxidants effectively attenuated that decrease in lung function. This is a really novel therapy. Antioxidants, basically helping individuals gain or have a less of a decrease in their lung function. So what does this bring us all to? This small review of all the literature is we want to identify what diesel exhaust does with regards to airway oxidative stress in healthy and mild asthmatics. And we're going to do this by looking at exhaled breath condensate levels of pH and 8-isoprostane. So from our literature review, we identified hypotheses that we wanted to test for. We identified that we hypothesized that diesel exhaust would cause airway oxidative stress as a measurable decrease in pH as well as an increase in 8-isoprostane. We also hypothesized that oral supplementation with N-acetylcysteine would mitigate these effects. And lastly, our literature review, although not mentioned in this intro, identified that sex and age were significant determinants of systemic oxidative stress and needed to be taken into consideration as potential effect modifiers in our study. And I'll explain the importance of effect modifiers in the next slide. One of the major pluses for our study, a very, very good strength, is its design. So it's basically a randomized control trial with 3 exposure conditions. Each and every one of the subjects participating in our study gets exposed to diesel exhaust with a placebo, diesel exhaust with an antioxidant, and fresh air with placebo. And this design helps us basically remove all confounding that could happen with individuals' biological variability. What it doesn't help us, though, is these factors, these covariates, are still potential effect modifiers in our study. So I don't know if most of you know what effect modification is, but sometimes what happens is you'll do a study and third variable will come in and modify the effects of your outcome in a certain way, and I'll explain a little bit more of that later on. So basically, we expose individuals to 300 micrograms per meter cubed of particulate matter. Some of you might think that's quite high, you know, and if you're comparing to ambient levels of diesel exhaust, it is quite high to what you and I would normally breathe in a day in Vancouver. However, occupationally, this level is actually quite low. For individuals working in underground construction or, you know, underground mining, 300 micrograms per meter cubed is actually quite small. So we're in the mid-range. And for most controlled exposure studies, you know, 300 micrograms is well within the range of the concentrations that were used in those studies. For antioxidants, we supplemented with 600 milligrams 3 times daily for 5 days prior to the exposure. And the exposures were 2 hours long with 2 15-minute periods of light bicycle exercises. This was to keep the respiration rate somewhat constant throughout the study. If we didn't do this, they'd probably fall asleep. Exhaled breath condensate was obtained 2, 6, and 30 hours after exposure. We aliquoted each sample into 4 separate aliquots and froze them at -80 before analysis. We analyzed 8-isoprostane with our own in-house method that we developed here at UBC with high-pressure liquid chromatography and tandem mass spec, so courtesy of our old chemist Phil Provencher and Winnie Chu. And we were able to reach a limit detection of 1.44 picograms per ml. And I can talk more about that later if you guys are chemists and interested in that. For pH, a standard protocol, we deaerated with argon for 10 minutes at 350 mLs per minute. We used an Accumet pH meter with micro KCl probe and calibrated at two calibration points prior to every exhaled breath condensate pH measurement. So why is it important to deaerate with argon is that every exhaled breath condensate sample has CO2 in it already. But what happens is the environment, carbon dioxide, diffuses within the sample, creating HCO3, thereby a weak acid acidifying your sample. So that just biases your reading. So what you want to do is you want to purge all the carbon dioxide from your sample so as to not bias your readings and create a standardized readout of your pH. And this little diagram is just my setup for measuring, for deaerating with argon, after which we'd measure pH. So what do we end up with? All this talk, we end up with 2 databases of biomarker levels at 0, 2, 6, and 30 hours after exposure. We then created 2 other databases where we corrected our values to baseline. So what you see in the top part of this graph is just the 8-isoprostane pH time points, all the data we have for that, and then the corrected time points that we have below. We want to correct for baseline just to see if, just to correct to make it a standardized measurement. So baseline levels, if they come in with, a certain amount of oxidative stress and then they increase, you want to make sure that you account, the change that you're looking at accounts for the baseline levels. What we hadn't necessarily anticipated before looking at the delta levels was the fact that although we controlled so much within our study, we couldn't necessarily control the environment they were in before they came into the study. So this makes maybe a little bit of bias in our baseline levels with regards to the external environment that we were not able to control for. And so that's why it's also important to look at post-exposure levels and not simply delta levels. For the process, or for the sake of this presentation today, I will only show you the first row here, so 0, 2, and 6, 30 hours after exposure, just because deltas are actually a little bit more complicated to explain and understand because you're looking at changes versus just directly looking at levels of 8-isoprostane or pH. But I can go into them afterwards if anyone's interested. We use linear mixed effects models to assess changes or effects of treatment on our outcome variables. For those of you that are statistician buffs, linear mixed effects models are basically a glorified way of saying repeated measures ANOVA. So what we ended up doing was we have our fixed effects from our dependent or independent variables, but we also had a random effect from the subject ID that we just randomly assigned our subjects with an ID. So we had to account for the types of variables we had in our study. We first looked at treatment overall effect on our biomarker levels, and after which we looked at the stratified effect, or the effect of treatment stratified by sex and age. So we looked at what treatment did for males and females, and then we looked at what treatment did for age 1, age 2, and age 3 individuals. After that was done, if we noticed a difference, we looked at interaction terms within our linear mixed effects model, and if that was significant. Personally, because I found the output so difficult to understand from interaction terms from the linear mixed effects model, I also did paired t-test to make sure that my interpretation was sound and correct. So that's what I'll be showing today. We also did multiple covariate models, but again, for purpose of time, I am not going to go through those results, just so I can show you the basic results of our thesis, of our project, and not go into too much detail. Finally, results. So our two databases, we had 23 participants for 8-isoprostane, and then we had 17 participants for our exhaled breath condensate pH database. And what you see is that the population is pretty young. We have a mean age of about 30 years old. The BMIs are pretty normal, so in the normal range. And so for Vancouver, being young and healthy is quite average. We have, well, it's average to be young and healthy. I don't know, I'm sorry.
It's a reasonable representation of the Vancouver population.
It's a reasonable representation of the Vancouver population. Thank you, Ed. So there was approximately the same amount of males and females, maybe a little bit more males than females. And I put the methacholine response status and atopic status in this table just for added information, but I won't go into the results of those covariates in this presentation. But it was interesting to look at the different proportion of individuals in both data sets. So 8-isoprostane with the 23 individuals had 14.5% of our data was below the limit of detection, which we decided to substitute with the LOD over square root 2 method. As you can imagine, the distribution was log normal, so we transformed it in order to perform parametric analyses. For pH, we had 17 participants. 6 subjects had multiple missing time points, with a lot of those being key time points we wanted to look at, and so we had to exclude them. But overall, pH was normally distributed. So I know this is kind of daunting as an image to look at. There's a lot to look at, so I'll take you through it quite slowly. So what you're looking at are the exhaled breath condensate levels of 8-isoprostane. The top left corner is the baseline levels. The top left corner is baseline levels. The top right is 2 hours after exposure. Here we have 6 hours after exposure on the bottom left, and we have 30 hours after exposure on the bottom right. And what you're seeing is logged levels on the y-axis, and on the x-axis are the different exposures. So we see fresh air with placebo, diesel exhaust with placebo, followed by diesel exhaust with antioxidant supplementation. Excuse me. I think what's important to look at here is the trends. So what you end up seeing is that from fresh air to diesel exhaust for 2 hours, 6, and 30 hours after exposure, we see an increase from fresh air with placebo to diesel exhaust with placebo. So we see an increase in, small increase in 8-isoprostane levels. At 2 and 30 hours after exposure, we see that this increase decreases back to initial levels, or a little bit lower after antioxidant supplementation. And so basically what we can see is none of the results were significant, unfortunately, but there are trends that we can look at. And significance isn't everything. Trends are actually pretty interesting when we're looking at overall levels. And the boxes, before I go on to the next slide, the boxes represent 50% of the population. The line within the box is the median, and the red line linking all 3 exposures together is the mean. And the whiskers represent the max and the minimum of the distribution, just so you can understand where the variability lies. I think what's interesting to see is that at baseline as well, levels were not constant. We didn't have the same amount of 8-isoprostane at baseline for all 3 exposures. So that could be just an artifact, again, of the outside So the outside world to our study was actually affecting the baseline levels of oxidative or 8-isoprostane. For pH, the changes are actually quite smaller. So we do see at 2 and 6 hours after exposure a slight acidification from fresh air to diesel. However, 30 hours after exposure, there seems to be really no change between the two. And there's maybe a slight increase from diesel exhaust to antioxidant 2 hours after exposure, but really we don't see much change across the board for overall 8-isoprostane, or sorry, overall pH results. Again, none of the results were significant. So as I mentioned, we went through stratified analyses by sex, and we identified that 6 hours after exposure there was a difference between males and females. And when we delved deeper into that by looking at the interaction term between treatment and sex, we saw that it was significant at 0.03 p-value. And then when running paired t-tests, we saw that males significantly showed a significant acidification after diesel exhaust exposure from fresh air, and then had their levels return somewhat to normal after antioxidant supplementation, but this was not significant. Another interesting thing is for females, we didn't really see the same type of trend. We actually saw the opposite. So we saw an increase in pH after diesel exhaust exposure compared to fresh air, and we saw an acidification from a significant acidification from diesel exhaust with placebo to diesel exhaust with antioxidant. And I'll discuss the meaning of those results in the discussion. Lastly, we looked at age, and age interaction term was significant 30 hours after exposure for 8-isoprostane levels. And what we found was actually pretty interesting. We saw the most significant results from our paired t-tests identified that for our age 3 category individuals, or 35 plus individuals, they had a borderline significant recovery in 8-isoprostane levels. So we went from a higher diesel exhaust, or a higher 8-isoprostane level after diesel exhaust exposure to a lower level after antioxidant supplementation. What's also interesting in this graph is that age 1 and age 3 individuals have the similar types of trends. So they both start at the similar level at fresh air and placebo, both increase after diesel exhaust with placebo, and both show slightly decreased levels of 8-isoprostane after antioxidant supplementation, although not necessarily significant for age 1 individuals. So overall, this is just a repeat of the results. As you all know, significance is something that we strive to achieve as researchers, but oftentimes is a little bit overrated. So just because these results were not necessarily significant doesn't mean that they're not actually showing something interesting. So I'll discuss why that is in the next couple slides. But what we saw was that diesel exhaust non-significantly increased 8-isoprostane levels, as well as non-significantly lowered pH after exposure with placebo, with diesel exhaust and placebo, and that these levels return to somewhat normal levels after antioxidant supplementation. Again, for the secondary analyses, males significantly acidified pH 6 hours after exposure, while older individuals showed decreased levels of 8-isoprostane after antioxidant supplementation. So what this might mean for a public health significance would be that, in our literature, what I think is really interesting is in our literature review, we identified that pH was not different in males and females. However, this study shows that after exposures to diesel exhaust, there is a difference between males and females in the way they react to diesel exhaust inhalation. So what this might mean is for any individual working in policy or in occupational health that's interested in the effects of diesel exhaust, males might be more at risk of having oxidative stress in their lungs after diesel exhaust exposure. What that means long term is still somewhat unclear, but it is something to be further investigated. Another interesting component is several studies showed that with increasing age bracket came increasing 8-isoprostane levels, And our study identified, we didn't see that in our study. We actually saw that for younger individuals and older individuals in our study, there was no significant difference between the age groups. And so they all pretty much acted in the same way, if I can say so. And older individuals had a larger decrease in atazoprostane levels after antioxidant treatment. So this might mean that older individuals react better to antioxidant supplementation than certain individuals that are 20 years or 30 years of age. So if you're 40 years old, perhaps antioxidant supplementation works better for you. But this, again, comes with a lot of caveats. But before I get into the limitations of that interpretation, I'm just going to discuss the main difference in our pH results with McCreener 2007. So McCreener was able to show a significant acidification after Oxford Street exposure when compared to Hyde Park. And we hypothesized that the difference between why we weren't able to show as big of a significance as they did was twofold. Their study population was mostly moderate and mild asthmatics, whereas ours were mostly healthy and mild asthmatics. And when you delve into the literature or delve into the study a little bit more, you see that the moderate asthmatics are pulling the significance of this association that we went through in the beginning. Also, what could be going on here is that the exposure in Oxford Street and in Hyde Park, they weren't necessarily only to diesel exhaust like we did in our study. They also had allergens and ozone exposure, and there is a lot of literature out there that shows that concurrent exposures for diesel exhaust and allergens together have enhanced health effects than just the two separated. Same for diesel exhaust and ozone. The two exposures together have higher respiratory health effects than do the two exposures alone. So the limitations in our study. So we were able to, one of the biomarkers that we decided to look at was atazoprostane. But what we ended up finding is that most the results were close to or somewhat close to the limit of detection. And what happens when your levels are close to the limit of detection is there's more systematic error with regards to your program and how those concentrations are calculated. So the closer you are, the more variability there is. And this basically contributed to having our probably not being able to see as many significant differences because of this inherent variability. There's also the issue of multiple comparisons. So when you're doing, let's say, 100 statistical tests, you do have a probability that 5 of those tests, if they're significant, will be by random chance. And in order to stay away from this possibility that we are seeing results that are just due out of random chance, the results that were significant in both single covariate models as well as multiple covariate models were given a bit more confidence than the results that were just significant in either or statistical tests. So although I didn't show the results from the multiple covariate models today, in the final report, those results that were significant in both in both types of tests are actually given a bit more weight than the ones that were only significant in one type of statistical test. There's always the issue of outliers. So because we have individual level data, we have to look at, is the significance being pulled by only one individual who's an outlier? And what we were able to do is look at every single data point and make sure that it was not an outlier. So do a Bland-Altman test, for example, to identify concentrations that were 2 standard deviations away from the mean of the population. And if those concentrations were found to be outside 2 standard deviations away from the mean, we then look deeper into that result to see if, is there something weird going on here? Is it an actual result, or is it just a product of chance that it isn't within the study, within the mean? And I have results at the end of this presentation that I can show with regards to that. Last but not least, to show statistical significance, we originally calculated that we needed 27 participants in order to have a full sample size. And as happens in several studies, you lose subjects along the way, or samples get contaminated. Again, there's outliers, or You couldn't collect the sample from the participant. So what ended up happening is we only had 23 subjects for azoprostane and 17 for pH. So that's also perhaps why we didn't see as much statistical significance in the primary analyses than we would have liked. And I think that concludes it for me. I'd like to thank WorkSafe BC for the funding, Chris Carlston, Karen Bartlett, and Mike Beller for their immense help during this process. Again, special thanks to Phil Provencher and Winnie Chu for helping me develop the azoprostane method. Here are my references, and I will take any questions.
Well, very impressive work, Bianca. Congratulations.
Thanks, Jeff.
I was wondering if we could just talk a little bit more about that McCreener paper. So if I got it right, they sent people to walk around, was it in London, you're saying, in Hyde Park? Yep. So do you have any idea what the diesel particulate levels are, usually on a typical London day? I mean, it's probably not as high as what you were pumping into that chamber of people, is it?
You know, I know they published this in their study. I'm pretty sure they gave us the overall exposure to diesel that these individuals had, but off the top of my head, I wouldn't be able to answer. I think it was less than 100 micrograms per meter cubed, I'm pretty sure, but I'm not 100% sure.
Yeah, sorry to put you on the spot about that.
No, that's great. I should.
That figure. But I mean, that seems pretty significant to me that they weren't controlling, or there were plenty of other airborne contaminants that could have caused this response. So I'm not sure where I'm going with that. I just seems like awesome.
30 micrograms per cubic meter in Hyde Park, but they make the case, the title of the paper has diesel exhaust in it, and they make the case that it's pure diesel exhaust, but so it's true that that street, Oxford Street, is 90-something% diesel traffic because of the way it's set up, so that is true, but it's also true, so the particles are probably coming mostly from diesel vehicles, that's true, but it's also true, as Bianca said, that there's other stuff in the air. So while the particles, the PM, are mostly from diesel traffic, it's also true that there are gases and other things like allergens. So, but 30 would be high for Vancouver, quite high. But it's that particular street, which is why they chose it, is one of the highest pollution urban streets in London. So.
Yeah, I got a question or two. I'm involved with people that are working on underground construction. And so they're exposed to, well, rock dust, some of them, and diesel exhaust. Have you got any suggestion as to if any of them or all of them should be using antioxidants to try and offset the effects of their exposure to these things?
Well, I mean, unfortunately, I don't think I've been able to show that antioxidant supplementation is for sure like helping these individuals against diesel exhaust exposure. I mean, personally, I think anyone should be taking vitamins every day just for the normal beneficial effects they have. And if by any chance it helps them against these exposures, then yes, I think they probably should be. Especially if they're breathing in, double types of respiratory particulate matter. I think that might help them.
Right, yeah, and these people are typically exposed 11 hours a day, 7 days a week. But
they have respirators?
Nope. No, that's traditionally, and in underground mining, it's the same thing, right? That people are routinely exposed for fairly long days, and they work 3 weeks in, 3 weeks out kind of thing. Yeah. In a lot of locations. So yeah, this has relevance certainly for those folks.
Yeah. Sure.
Two things about antioxidants. One is that in general, there's sort of a no harm or little harm philosophy, such that people, if they want to take them, workers or even non-workers, it's probably okay. But there are a couple caveats. One is that at high doses, like it's sort of the axiom of life in general, anything in excess is probably bad. And it's true actually that at high doses, antioxidants have been shown to have adverse effects, which I won't go into further. So some people at the extremes who want to just shovel antioxidants down in excess could actually suffer adverse consequences. And the other thing is, of course, as everyone here I'm sure knows, we don't want to ever sort of mask the primary root problem where we're trying to aim for lower exposures by giving people antioxidants and saying, well, you know, sorry you don't have a mask, but at least you can take these pills. I know you weren't suggesting that, but just for in case anyone, because sometimes, you know, people only hear a snippet of things and, oh, remember, you know, Ed was talking about giving pills to these guys. And I know you didn't mean that, but we just have to be, you know, extremely clear that that's not a good idea.
Yes, reducing exposure is obviously where I come from. And the progress is being made in that area, yeah.
But I do know that you don't particularly think that slapping a mask on is better than ventilation. Thanks very much, Bianca. I'm so glad you got to present. And I wanted to ask you, you talked a lot about the issue of statistical significance. I can't just let it go by, but I mean, one of the possibilities that you alluded to is that the power is low because you had fewer subjects than you had originally planned. But the other possibility, although you saw differences, is that those differences were very tiny. And
the effect size.
Yeah, the effect size looked small. I mean, it's hard for me to know. So that's what I wanted you to comment on. Were the changes that you saw the kind of changes that you thought were clinically important, and therefore if you had power you would have thought, yes, this is an important change?
Yeah, that's a great point, and I'm glad you brought that up. The changes, so what Kay brought up is actually really important. You know, is there a decrease in pH, or is there an acidification? If so, is the amount of acidification, you know, important to look at or not? Or same for idazoprostane. I think clinically, yes. I think the changes, unfortunately, if they weren't significant, it's, you know, it's too bad because of the power issue. But personally, looking at this data and looking at the changes in idazoprostane level, I think they're large enough to identify that yes, there is a significant decrease, and large enough to elicit that if we had more individuals, we would see statistical significance. And if this is important clinically, I mean, a change of, an average change of, let's say, 50 picograms per ml of idazoprostane, I think, is large enough to elicit that, yes, they are undergoing significant oxidative stress than what we would normally undergo. And this has been linked to DNA damage. I don't know the long-term chronic effects of oxidative stress on the lungs, but I'm pretty sure that that type of short-term damage, although probably quickly repaired, can, in the long term, be detrimental. So with regards to, I guess, the results presented today, yeah, I would definitely say that clinically, I think what was shown today, although not significant, is important to take into consideration. But thank you for that question.
Bianca, question for me. Thanks for an interesting talk. You made reference, and I think I heard you correctly, that this influential paper, McCreener, their subjects were, many of them are moderate asthmatics, mild, moderate asthmatics, whereas your study, you had the more milder asthmatics, right?
Yep.
So the question is, in your subject recruitment, was it just by chance that you had mild rather than some combination of mild, moderate, such that you are not able to make a comparison with the McCreener results? That's my first question. And second is, were the severity of the asthma, the state of the asthmatics, were they diagnosed at some point to be qualitatively so, or did they declare it themselves? Like, what made them mild?
Yeah, that's a great point. I didn't go into the asthmatic part of our study. We were recruiting about 1/3 healthy individuals and 2/3 mild asthmatics, so that was part of our recruitment process. We were not looking for severe asthmatics. We were looking for more to see if we could show a difference between those that were mildly asthmatics and healthy individuals. With regards to diagnosis, asthma is a complex syndrome that's somewhat difficult to diagnose, and so to put them in the box of mild asthmatics, typically they had to be responsive to methacholine at 8 milligrams per mLs of methacholine. So any concentration lower than that, they were shown to be responsive to. That was a criteria. Another criteria was, I think they had to be at least diagnosed at some part in their lives with asthma. It wasn't necessarily a clear diagnosis, like did you have asthma at any point in time was one of the questions asked. Also, ATP was part of our diagnosis. So there are several components going into the diagnosis, or the classification of these individuals into that asthma type, particularly only using their inhaled corticosteroids, when really necessary, if not at all. I think, Chris, if you can comment a little bit more on that characterization.
Basically, the approach was to try to pull people in based on their not self-diagnosed, but their self-reported asthma, that a doctor had told them that they had asthma, and then we used our own methodology to confirm whether that was true based on a thing called methacholine challenge. But in a nutshell, because of the crossover nature of the study, whether or not they had asthma by the absolute strict definition was not particularly limiting, because we also wanted to see how people with or without asthma responded. And so having a majority of asthmatics and then a few non-asthmatics allowed us to also make that contrast. In terms of the relationship to McCreener, the McCreener definition of asthma would be very similar to ours, which would be physician-diagnosed asthma. So I don't think, in terms of, I think the root of your question, Lydia, the differences between us and McCreener, I don't think are attributable to a dramatically different definition of asthma. I don't know the exact answer, but I think Bianca's hypothesis about co-exposures is as good as any other at this point.
I enjoy your presentation.
Thank you.
But one thing I'm wondering is, if you have, maybe I missed it in your presentation, if you have individuals exposed to fresh air with the antioxidant, how does their A-tyrosine level compare to individuals exposed to fresh air with placebo? And maybe if you compare those individuals with fresh air and antioxidant to your diesel exposure with placebo, maybe there will be a bigger difference. That's what I'm wondering.
Oh, well, that's an interesting question and comment. Our study, unfortunately, we weren't really interested in just the practical aspects of antioxidant treatment to see if it changed the oxidative stress levels after fresh air exposure. Our fresh air exposure with placebo was really just to be a control to see if diesel exhaust with placebo did anything at all. And the antioxidant part of the study was really just geared towards the diesel exhaust exposure section. So we didn't look at fresh air with antioxidant supplementation, unfortunately, but interesting.
Hi, Bianca. I have an online question, and still it's on antioxidants. And the question was, were the antioxidants supplied restricted to N-acetyl, or were there other antioxidants supplied?
Also a good question. So in our study, we supplemented with only N-acetylcysteine, and we asked the individuals to control their consumption for our exposure day, so to only eat the same thing every time they came in, just to make sure that if there were other antioxidants present, that they were controlled for at every exposure day. So there probably were other antioxidants within those individuals' personal diets, but those were controlled for in our study.
Another question that occurred to me is, did you ask about exposures they might have had to diesel exhaust between the 2-hour period in your test chamber and the 30 hours post-test?
That's a good question. We didn't actually ask those questions at all, no. We asked, basically we would check between the exposure day and the post 30 hours was if they had any adverse events, so if they had experienced any respiratory issues or, but not diesel exhaust. We didn't ask that, no. That's a good question. Probably should have.
Just, okay, follow up to that. Did any of these people work in an environment like a maintenance garage or something of that kind where there was routine exposure to diesel exhaust?
To my knowledge, no. To my knowledge, these were regular, everyday people that were not necessarily heavily exposed to diesel exhaust.
No occupational exposure that you
yeah. Not that I would specifically point out and go, ooh, we can't.
This guy's a mechanic. Yeah, he's a mechanic, right?
Yeah.
Okay.
A good question.
You mentioned that there was an effect modification between the sexes, and I was wondering if there was a proportionately higher sex in each of the age group that sort of made the other trend that you found with age, like it's not all linear.
Oh, you mean like just if age 3 only had males kind of thing?
Yeah, or like only one female and the rest are males and the rest are all just females.
That's a good point. It's a great point actually. So in these kinds of studies, you want to make sure that the results you're looking at aren't, the effect of something else, right? So what we ended up doing was, I can assure you that age is not an issue sex is not an issue for adazoprostane levels. And so when it comes to looking at the age effect in adazoprostane levels, it's not necessarily modified by sex, but if it were, a multiple covariate model would control for that. So having those covariates in a model when you're controlling for sex and you're looking at the effects of age, well then your model effectively controls for age in your, effectively controls for sex in your age result. So, but pH only seemed to significantly affect, well, from what I showed, in males. So that's separate from adazoprostane. So their relationship would be interesting to look at, I guess, between the two. Good question, Ivan.
Thanks for that, Bianca. Congrats on all your hard work.
Thanks, Billy.
You mentioned very briefly, you also had your analysis done on the differences, so on the delta values. I'm just wondering if you just very briefly summarize if you saw essentially the same thing or what you saw there.
To a lesser extent, yes. So one of the main issues with our delta values is that the variability within the deltas are very high. So seeing the same thing in our post-exposure models than our delta levels was a little bit more muddled, I guess. The results were just, there was a lot more variation in the deltas than there was in the after exposure because of the baseline levels. We did see certain results come back multiple times. For example, 2 and 6 hours after exposures were typically the time points that we saw the most acidification. 6, for example, came back, methacholine, other covariates that I didn't show today, they also were significant in those models. But that's a great question. I wish I brought a slide on that, but I did not add that slide. Thanks, Billy.
Have you considered doing a study on people that had, say, more than 5 years occupational exposure to diesel exhaust at higher levels, obviously?
I think our colleague Mandy is going to be starting does her study take into account the exposure? I think she's looking at, not measuring, qualitative. So our colleague is doing a qualitative study on I don't know if it's the BC ferry workers or if it's changed, but she's.
Yeah, it's a bunch of workers that's really about their
perception of exposure. Okay. Well, then, so the answer is we didn't do that, and
no,
unfortunately, our
just a thought for the future.
It's good. Yeah, our lab is very experimentally driven. So with regards, there's always opportunities to open that up to thesis students if they they would be interested in working with Chris with that, that's definitely a possibility. That's it. Yay.
Yes. Thanks, Bianca, for an excellent piece of research work and a good report on it.